Now, welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that the spindle assembly checkpoint is actually ensuring the bipolar attachment of all of the pairs of the sister chromatids to the opposite poles. Now, how the spindle assembly checkpoint is going to check that I told you that after the replication of the DNA in the S phase, all of the sister chromatids, they are actually attached to each other by the cohesion proteins, and when these kinetochore microtubules they want to pull the sister chromatids to the opposite poles, there is actually a tension on these kinetochores. Now, if this tension is present on all of the kinetochores, on all of the pairs of the sister chromatids, that means that everything is okay. You can actually move into the next phase of the cell cycle, that is the anaphase. Now, if there is not even a single kinetochore that is unattached, then the spindle assembly checkpoint is going to stop the cell cycle at the metaphase, and therefore not allowing it to move into the anaphase. And it was achieving this by that the unattached kinetochores, they were attached by a special protein that was known as the MAD1 protein, and this MAD1 was part of the spindle assembly checkpoint. This MAD1, after attaching to the unattached kinetochore, was actually going for the activation of the MAD2 into its active form, which was the c-MAD2. Now, the c-MAD2 that was interacting with an important protein, the CDC20, thereby making it inactive. And if the CDC20 that is inactive, that means it cannot activate a substrate, which was the Anaphase Promoting Complex. So the Anaphase Promoting Complex was in the inactive form. You actually cannot move into the anaphase of the cell cycle. Now, when all of the pairs of the sister chromatids, they have got tension on all of the kinetochores, that means that there will be no unattached kinetochore, and if there is no unattached kinetochore, that means that the MAD1 protein cannot interact with the kinetochores. Therefore, the c-MAD2 will not be going to get activated. That means that the CDC20 will not be in interaction with the MAD2. That means that the CDC20 will be in the active form, and it can actually go for the activation of the Anaphase Promoting Complex. Now, let us discuss the functions of the Anaphase Promoting Complex, how this Anaphase Promoting Complex is actually promoting the anaphase. Now, this CDC20, as I've told you, when that is activated, that is going to bind to this Anaphase Promoting Complex, and the Anaphase Promoting Complex is going to get activated. This Anaphase Promoting Complex, the APC, is also called as the cyclosome, and this Anaphase Promoting Complex is a large protein complex made up of about 11 to 13 subunit, and this Anaphase Promoting Complex is actually in E3 ubiquitin ligase. I'll be having a detailed discussion on the ubiquitin proteasome system in my next video, but for this particular time, keep in mind that the E3 ubiquitin ligase is one of the enzymes that is responsible for the attachment of the ubiquitin protein to its target protein, and when it attach a ubiquitin protein to its target protein, that particular protein is degraded by the proteasome. So, when the Anaphase Promoting Complex, when it is activated, what are its functions then? One of its important function is the activation of an enzyme which is known as the separase enzyme. Now, I told you that the sister chromatids, they are attached to each other by these cohesion proteins, and when the kinetochore microtubule, they want to pull the sister chromatid to the opposite poles, tension is created on these kinetochores, and you cannot separate the sister chromatids from each other until you have degraded the cohesions. So for the degradation of the cohesion, the specific enzyme is the separase enzyme. The problem is that this separase enzyme is attached to its inhibitor, that is another protein which is known as the Securin. Now, what the Anaphase Promoting Complex do, that it is going to attach a ubiquitin protein to this Securin, and this Securin is going to be degraded by the proteasome. So when the Securin is degraded, that means that the separase will be activated, and when it is activated, that means it is going for the degradation of the cohesion protein, and the kinetochore microtubule can actually separate the sister chromatids from each other and move them to the opposite poles. If you talk specifically about which subunit of the cohesion complex is degraded by the separase, that is the SCC1 subunit, or that is also known as the glycine subunit. Now, if you remember, I've told you that this cohesion is a protein complex, and out of this protein complex, the separase enzyme is specifically going to degrade this SCC1 or the glycine subunit, and when this important subunit is degraded, whole assembly of the cohesion protein is going to be removed from the sister chromatids, and the sister chromatids can easily be moved to the opposite poles. And you can actually see it over here that when you have degraded the cohesion proteins, these microtubules can actually move these sister chromatids to the opposite pole. This member of this pair is going to move to this particular pole, and the other member is going to move to the opposite pole. Thereby you can actually make two clear poles with chromatids present on them. Another important function of the Anaphase Promoting Complex is that it is going for the degradation of the cyclin B in the same manner that it is degrading the Securin protein. It is going to attach a ubiquitin protein to the cyclin B, and the cyclin B is going to get degraded by the proteasome. Now, when this cyclin B is degraded, that means that your MCDK is going to get inactivated. If you remember, the MCDK was a complex of the CDK1 and the cyclin B, and is the CDKs, they are the cyclin dependent kinases, and if you degrade their respective cyclin, those particular CDKs, they get inactivated. So once you have degraded the cyclin B, that means your MCDK is going to get inactivated. Now, what will be the advantage if you are going for the degradation of the cyclin B and the inactivation of the MCDK? Now, if you remember, this MCDK was performing a lot of important function at the start of the M phase. One of its important function was the condensation of the genetic material. Now, if you have separated the chromatids from each other and they are now on the opposite poles, and if they are still in the condensed form, that means that they will be transcriptionally inactive. And after the separation, we do not want these chromatids to be transcriptionally inactive. We want them to be transcriptionally active, because after the cytokinesis, you are going to get two cells, and if the genetic material is transcriptionally not active, that particular cell is not going to survive. But you cannot make these genetic material transcriptionally active unless you have made them in the loose form. You have removed the condensation of the genetic material. If the MCDK is activated all the time, that means that the condensation complex, that will be active all of the time, and because of the condensation complex, the genetic material will always be in the condensed form. So once you have inactivated the MCDK, that means that the condensation complex is going to get inactivated, and if the condensation complex is inactivated, that means your genetic material is going into the loose transcriptionally active form. That is why we are actually going for the inactivation of the MCDK by the degradation of the cyclin B. Another important function of the MCDK was the elongation of the microtubules by the activation of the GAF proteins. Now, look at over here. If these microtubules, if they are always in the elongated form, that means they will not be able to move the chromatids to the opposite poles. This MCDK, as it was responsible for the elongation of the microtubules, so once you have inactivated the MCDK, that means that the microtubule, they are going to shrink, and when they shrink, they are actually moving the chromatids to the opposite poles. Another important function of the MCDK was the breakdown of the nuclear envelope. Once you have separated the chromatids from each other, the other important thing at the telophase you have to do is that you have to make the nuclear envelope again. if you are not able to make these nuclear envelopes again, like you have separated the chromatids from each other, the next important thing, the telophase, is that you are going to make this nuclear envelope around this. So if you are not going for the inactivation of the MCDK, that means your nuclear envelope will always be in the degraded form, and you will not be able to make the nuclear envelope around the two new nuclei that you have just made. But if you have inactivated the MCDK, that means that the nuclear envelope, they can be made again. That is another important function of the Anaphase Promoting Complex, that by going to degrade the cyclin B, it is going for the inactivation of the MCDK, thereby reversing all of the functions of the MCDK. Another important thing that the Anaphase Promoting Complex is doing, that it is going for the inactivation of the SCDK. And if you remember, the SCDK was a complex of the cyclin A and the CDK2. And this SCDK was actually responsible for the phosphorylation of the origin recognition complex, thereby ensuring that in one cell division, the DNA replication gets only once. So if you are going for the degradation of the cyclin A, that means your SCDK is going to get deactivated, and when that is deactivated, this phosphate group will be removed by the phosphatases. And once you have removed this phosphate group from the origin recognition complex, that means that in the next cell cycle you can actually go for the replication of the DNA. So if you are not going to degrade this phosphate group will be stuck to the origin recognition complex all the time, and after the cell division you will not be able to go for the replication and hence for the division of the cell. So by degrading the cyclin A, the SCDK gets inactivated. The phosphatases, they are going to remove this phosphate group from the origin recognition complex, hence you can actually go for the cell division, and you are actually preparing yourself for the next cell division. Another important thing the Anaphase Promoting Complex is doing, that it is going for the degradation of the germinin protein. If you remember, I've told you that the SCDK, that is going for the phosphorylation of the origin recognition complex and also for the degradation of the CDC6. By phosphorylating the CDC6 was degraded by the ubiquitin proteasome system. But the CDT1, that was actually inhibited by its interaction with another protein, that was the germinin. Now the Anaphase Promoting Complex, it actually go for the degradation of the germinin in a ubiquitin proteasome dependent manner, and when the germinin that is degraded, that means that the CDT1 is now available for the next round of the cell cycle. Once you have once the Anaphase Promoting Complex have done all of its function, that means you have gone for the contraction. You can go for the contraction of the microtubules. You have contracted the microtubules. The nuclear envelope that has been made again, that means you have now two nuclei present in a very big cell, and this phase is actually called as the telophase of the cell cycle. You have made the nuclear envelope around the chromatids that you have separated from each other by the contraction of the microtubules. Now if you remember, I've told you that the M phase is actually divided into two sub phases. One was the M phase, the mitosis phase. So but at the end of the telophase, we are actually done with the mitotic phase of the M phase, and that is we have divided the nucleus. We have got for the nuclear division. The next phase, which is the cytokinesis, and in the cytokinesis what you are going to do is that you are actually going for the separation of the two nuclei from each other. Now this function is actually achieved by a contractile ring. Now this contractile ring, it is made up of the actin filaments, the myosin-ii and its associated protein, and this contractile ring is going to make a cleavage furrow about in the middle of the cell, and this contractile ring is actually going to deep this cleavage furrow, and when this cleavage furrow deepens, actually the two daughter cells, they are going to get separated from each other, and these two daughter cells, they are actually identical to their parent cells. Once you have gone for the cytokinesis, you have made two identical daughter cells. What is going to happen next? Now, if the conditions they are favorable, by that I mean that if the nutrients are available, the growth factors they are available, these growth factors are going to bind to the growth factor receptors present on these daughter cells, and when these growth factors they bind to their growth factor receptors, gene expression is going to change in these daughter cells, and when the gene expression that is changing, that means that the cell will move from the M phase again into the G1 phase, and it will be going for the division again. If the conditions they are not favorable, that means the growth factors they are not available, that means they are not going to bind to the growth factor receptors, the cell is going to move into the G0 phase. It is going to perform all of its function, except it is not going to divide. We'll continue this in the next video.